Welcome everyone to our seminar on generative AI in drug discovery that is generously sponsored by VantAI. I'm Michael Bronstein. I'm the DeepMind chair of AI at the University of Oxford and also chief scientist at Vant. And today we have the great pleasure to have the talk given by Guillaume Huguet and James Vuckovic, both from Dreamfold. So Guillaume is a PhD student at Mila. And James is the senior machine learning researcher at Dreamfold. And they are going to talk about the latest and greatest advances in FoldFlow, which is a sequence-augmented SE3 flow matching algorithm for conditional protein backbone generation. So this was a preprint that was published a couple of months ago. And without further ado, guys, thank you very much for joining. It's a great pleasure to have you here, and the floor is yours. So I will stop sharing here and allow you to share your slides.
Thanks for having us. Okay. Yep. Can I just request control?
Hello, everyone, and thanks for the invitation and introduction. So yeah, I'm Guillaume, and I'm with James today. We're going to present FoldFlow-2, a sequence-augmented SE3 flow matching model for conditional generation. And this is work done at Dreamfold with all the team here present, and we'll talk about them again later. Okay, so just a quick presentation of the a quick outline of the presentation. We'll start with a background, a problem setup, a quick review of flow matching and protein representation, a quick review also on FoldFlow, the first model. Then we'll follow up with FoldFlow-2, the training, the architecture, and we'll present our experiment on unconditional generation and conditional generation. And we have a few slides on our next steps and conclusion.
Okay, so just before we get into the background section, some motivating questions that guided us during this research and that hopefully we'll touch on in this talk. So the first one is we wanted to basically see how far we can push the success of these SE3 flow matching models like FoldFlow. So that was our initial starting point. We also wanted to better understand how we can guide generative protein models to do useful things, because if they can't be conditioned properly, it provides limited use to us. And then also, we wanted to see if we took the best practices in models, in data, in training, could we achieve really good performance in protein backbone generation? So those are the three areas that we tried to explore, and hopefully we'll be able to shed some light on each of these questions. So Guillaume, do you want to go into the background?
Yeah, so following with the background, the first thing we want to consider or to answer is what are proteins? And here it's going to be like an introduction of protein from an ML perspective, so not from biologist, but really like what tools or what data are we dealing with? So first, yeah, protein is a large, complex molecule. It plays a crucial role in the structure and function of living organism. And they're composed of smaller units, so amino acids, which form together a chain and then are represented either like as a sequence or a 3D structure like this. And it's worth noting that each of these amino acids are composed of different atoms. So here we have like a few examples of amino acid and the composition of their atom and their bond. Okay, and the main modality we observed, so this is the data taken from the PDB, the protein data bank, is this chain of amino acids and 3D coordinate of the atoms. So the amino acid sequence represented here in this PDB file, and the atom 3D position represented here. Okay, Okay, so here we can see like each residue. So this one, for example, starts at residue 26. So this residue has a few atoms, and for each atom, we have the 3D coordinate. And so the main, let's say the main protein model, the first generation, if we can say like this. So we'll just quickly review them. So one of the main one people know is the AlphaFold-2 and ESM-Fold. And the task here, or the data they deal with, is you take as input a sequence and you output a structure. So you learn to fold a sequence of amino acid into the 3D coordinate of the atoms. There's the other task, the inverse folding. So now you start with a structure and you try to predict the sequence of amino acid that could have folded into this structure. So two known models in this area is the protein MPNN and ESM-Fold inverse folding. And so again, it was often the case that we either go from one modality to another, or in the generation, it was using a modality to generate another modality. So you train on structure and you generate structure. So model like RF diffusion, FoldFlow, Genie, and on sequence. So you train on sequences, and at inference, you also generate sequences, model like Evotif. And the corresponding task, so in our case, FoldFlow, so we are here, and we still consider in this presentation a task of generation. So the end goal is that, like, at inference, I want to start from either like a mix of noise and structure and generate a new structure. And so one of the first tasks to consider is unconditional generation. If I just start from noise, can I generate protein that are novel or good? We'll define good later. Then another interesting task is motif scaffolding. This task can be considered either like in sequence space or in structure space. The main idea is that you want to fix part of your representation. So in this case, in the 3D structure, we fixed a motif, a functional motif, for example, and we want to design the other area of the protein. So in blue, this is the protein, the structure that we've generated. And in orange, this is the structure, the part of the structure that was fixed. And the same task applied also in sequence space. For example, this is taken from Evotif, a diffusion model for sequences, where they fix part of the sequence and they design the rest of the sequence. And this also follows the idea for binder design, where we want to fix a target and we want to create a protein from noise that will bind to this target. And so these are the main tasks we are interested in. And the paper will present mainly results on motif scaffolding, unconditional generation. And as a next step, the binder design task. Okay, so going back a bit to the data set, given this in mind, the task we want to consider, in FoldFlow, we worked on the structure space. So we mainly consider the atom position, and in FoldFlow-2, we incorporated the sequence information. So the idea for the atoms is like, if I have these two proteins, we see, well, or at least I'm telling you, they are the same, but they are rotated. So one of them is just, we apply the rotation to the initial one. And so for our model, in our case, like, we want to evaluate them in the same way. So we want these two proteins to be equivalent. And so there's multiple ways of doing this. One of them is following like AlphaFold-3 to do like data augmentation. So you augment your data set with some manipulation that will make this your model translation and rotation invariant. So for example, you will duplicate your protein with a few rotations. And in our case, we directly model our vector field network or just our predictive network on the space SC3. And SC3 is the space of rotations and translation. So we are equivalent to this space so that we don't have to consider data augmentation tricks. And so here is just a reminder that the data that we get is really one realization of this protein. So in a specific, like a spatial representation, but it could be also rotated or translated. So we have to do this little manipulation in order for the model to be equivalent, SC3 equivalent. So yeah, FoldFlow, we fall in this category and directly model on SC3. So yeah, it's a flow matching model designed on SC3. So the first thing we do is to create a representation of an instance of a protein so that it's translation and rotation invariant, so that the representation is translation and rotation invariant. And then we use flow matching. And I should just remind that in our case, we only model the backbone. So the backbone corresponds to these 4 atoms at every residues, but other atoms are present in this protein, namely the one for the side chains and other. But other models can consider these tasks. So I'll just review now, how do we represent this protein backbone? So the 4 atoms I had in the previous slide, how do we represent them and to feed them as input to our model? So if we have a protein with n amino acid, so we can think of a protein of size n, we want to represent it as n frames. So we have our fixed idealized coordinate, so the 4 atoms I mentioned previously, and we center them at 0 for now. Then for each residue, so residue, it's the combinations of multiple atoms. I represent it as a rotation and a translation. So the rotation component corresponds to my the translation component corresponds to my C alpha. And then given N and C, I can build a rotation matrices indicating me what is the relative position of these atoms with respect to my C alpha. So now with this representation, whether I do a translation or a rotation, the relative position of these atoms will be the same. So by building this representation, turning this atom coordinate into frames, this representation is invariant to rotation and translations. So yeah, if I decide to do a global translation, I would have the same representation. So this is the SC3 transformation, creating this tuple here. So now we know how to represent our data, convert it into frame. Now we'll just quickly review flow matching. We won't go too much into the details, but just to give the essential idea and how we train these kind of models. So maybe just a quick reminder on diffusion model, because they are closely related. The main idea of this diffusion model is we want to generate a given data distributions. And the idea is to first generate a data that is easy, a distribution that is easy to sample from, for example, a Gaussian distribution, and then denoise it progressively in order to turn it into our data distributions. And so we want to learn this process, turning data into noise, and then turning noise into data. And so in flow matching, it's similar, but we instead define this process as a deterministic process. So it's still generating distribution because the stochasticity comes from our initial and final distributions, but the path on the support is deterministic in this case. And so in our case, we really want to learn this vector field here, F, T, and X, so that we can sample from a Gaussian or a random protein and turn this protein progressively into the protein sampled from our data set. Okay, so slightly more details, but we'll say like this probability path. So how does the distribution evolve through time when it interpolates between these two? This, we say, it's caused or generated by a specific vector field. So if we have the vector field mu, which take as input time and our current position on our manifold, if we define a random variable following this vector field, for which the evolution is prescribed by this vector field, then we say that if x1 or xt, sorry, follow rho t, we say that mu t generates rho t. And so now we know that if we learn the specific mu t, we can generate random variables that will follow our target distributions, so or our data distributions. So what we want in flow matching is to learn a vector field as close as possible as this one here. However, it's impossible, or at least, yeah, it's impossible to learn directly mu t. And so the beauty of flow matching is to find that if instead of learning the unconditional one, you learn a conditional vector field here. So by fixing the two endpoints of your distribution, this vector field we can know and we can regress against. And the gradient of this loss would correspond to the gradient of a similar loss with the unconditional vector field. So in SC3 flow matching, the main difference is the metric here that we need to compute for a loss. We use the metric defined on SC3. And so this is the main idea how we train this model on SC3. So now James will follow with the FoldFlow-2.
Yep, thanks, Guillaume. Yeah, so that's the general setup. And then, okay, so before getting into the actual details, I just wanted to hopefully argue why sequence conditioning is a good idea. So as a general sort of statement, I think it's probably true that most generative models need to be conditioned or guided to do useful things. Like an image generator is not that useful unless you can tell it which images to generate. Same thing with text and same thing with proteins as well. For us, it's possible to guide structure-only models like FoldFlow. You can do the in-painting trick, you can use potentials to guide your flow, but it's true that protein sequences are highly informative about structure. In fact, they determine structure, as Guillaume mentioned earlier. And also, they are much more available than structure data. So we can approach larger scale machine learning problems if we are able to train on sequences. So given that, we think it is a good idea to use sequence information to condition a structure generative model. And so, if we do this, what capabilities do we gain? So you can think of parameterizing the different tasks by what the inputs look like. And we have now two different modalities of input, sequence and structure. And so if you fully mask the sequence and you pass in noise as the structure, that corresponds to unconditional generation, the same as FoldFlow-1 or a lot of different models. If you pass in the true sequence and noise and you ask the model to generate a structure, that is equivalent to folding. And so that's a new capability that we can get. Also, you can pass in partially masked sequences and structures, and that allows you to do in-painting or motif scaffolding by using conditional sequence inputs as well as structure. Also binder design, which is similar, except now you're masking one chain in, let's say, a complex, and you want to generate the other chain. And then also something that's very interesting is sort of partial folding, where you know, let's say one chain's sequence, but you want to simultaneously design a binder and fold the target at the same time. Well, this is something that you can do if you have this sort of two knobs to twist. Okay, so clearly there's a lot of good capabilities here. And so how do we go about actually doing this? Okay, so the architecture. This is the high level diagram that describes the FoldFlow-2 architecture. I'm going to go into each of these sections individually, but just at a high level, it's a pretty typical three-stage encode, process, decode setup from machine learning. Yeah, so nothing crazy there. It's inspired by and kind of shares similarities with folding models in the sense that you have a sequence path, you have a trunk, and then you have some sort of geometric decoder or structure module, as it's typically called in folding models. Except in folding models, there's no structure encoder because the task is specifically to take a sequence as input. And also our architecture doesn't use any recycles, which are these sort of iterative refinement steps of the output structure. And then also not shown here on this diagram, there's lots of normalization and stuff because, you know, combining different modalities together can introduce regularity issues. So for the encoder, basically, we have two different modules that encode the modalities separately. The structure encoder is an IPA transformer network. This is the same architecture that was used in FoldFlow-1, used in FrameFlow. I think it was also used in FrameDiff. It's a fairly well-studied and well-tested architecture for representing protein rigids. The encoder for sequence, as you can see, is an ESM2 model. I think we use the 650 million parameter variant. And that takes the sequence and produces some single and pair representations. And then the sequence to trunk module, which is shown on the right, is in a larger sort of zoomed in view, has different networks that are the same architecture as ESM fold for processing the single and pair representations into sort of the same, roughly the same latent structure as the structure encoder. And then for the trunk module, so we have the structure representations in pink and the sequence representations in green, and now we need to fuse them together into a single latent representation of the protein that knows about both modalities. And so the first thing we do is use a simple combiner module. It's really just a project using a linear layer and then concatenate together. Nothing fancy there. And then this sort of jointly embedded representation is processed by a number of trunk blocks. And these trunk blocks are the ESM variant of the evoformer. And the evoformer was a very successful architecture that was introduced in AlphaFold 2. And yeah, is another very well-studied sort of model component for building protein representation models. And so yeah, that's the fusion. And then at the end of the trunk blocks, you essentially have these representations for the single sort of protein and also the pairwise representations. Those are essentially enriched by the sequence of trunk blocks, and they should know about both modalities of the protein. And then for the decoder, so we have the outputs of the trunk module in blue, and we also have the skip connections from the IPA transformer in pink. And the IPA transformer at the encoder also produces single and pair representations, and it produces a rigid representation. And so we find that so we do some skip connections before passing to the structure decoder. The decoder is also an IPA transformer network. And yeah, that's basically, it's used to make the final prediction about the rigids that are used to derive the vector field. Yeah, one thing we found that was very important was these skip connections between the input or the structure encoder and the decoder. Mainly the intuition there is that the flow models, especially the decoder component, needs to have sort of clean time information to know at what point along the flow is it making a prediction. And the time information is encoded in the structure encoder. And so in order for that to sort of propagate clearly to the decoder, we needed the skip connections. So that was just something we learned along the way. Okay, so that's the basic setup. You have your protein structure and your sequence. They get encoded and then they get sort of fused and then they get decoded into a prediction of a vector field. And basically, we then generate proteins by starting from a noise distribution and then iteratively integrating the predicted vector field along a different number of time steps using Euler integration, like an Euler scheme for that. Yeah, there's just this sort of iterative feedback loop between the sort of current position and the predicted vector field. Yeah, one thing to note is that this sort of feedback loop only exists between the vector field and the structure. So you can actually cache this ESM2 call, which can be expensive. And so you only have to compute it once at the start of inference. And so that's quite powerful. Yep, that's how the inference works. And so then for training, so now we have this architecture that sort of processes sequences and structures, produces this vector field on SE3. How do we train it? Well, okay, so we start from a training example, which is a protein, right? And then the first thing you do is get the rigid for the data, which is x0. You sample your time, you sample your noise, and you compute the sort of true position and vector field along the flow that you want to regress your vector field against. And then you also get the sequence for that training example, and you sample basically whether you want to mask the entire sequence or leave it unchanged. And thus that happens with equal probability. And so then you have these two modalities as representations. You feed those to the FoldFlow-2 network in the exact way that we just described. And then you get a predicted vector field at the output, which of course depends on the parameters of your weights. And then you compute the loss. And we use the exact same loss as the original FoldFlow. And just at a high level, you can think of that as sort of a mean squared error, but done on the manifold SE3. And so there's a lot of kind of complicated math that goes into making that work well. And that was a main contribution of the original FoldFlow paper. We also use some auxiliary losses to augment that, but the main component is this SE3 loss. And then you compute the gradient of your loss on SE3, and then you use, Atom, whatever optimizer you want to update the network. And that's essentially one training iteration. So during our training, we mask 50%, so as I said, right, we with 50% probability, we're masking the entire sequence. Otherwise, we're leaving it unchanged. And so kind of implicitly, this means that 50% of the time we're training something similar to a folding task. And so we'll see later on that that kind of allows us to have pretty good performance on folding, even though we don't explicitly sort of try to do it. We also didn't really look at partial sequence masking, which would have been a sensible thing to do, but it's something we're investigating as a next step. Okay, so we have the loss and the sort of training setup. Let's talk about the training data for a second. We trained on a subset of PDB using relatively short, but not crazy short proteins, sort of maximum length 384, using sort of typical filtering that was described in FoldFlow and other papers. we also experimented with a larger subset of 186,000 structures using a combination of PDB and also the Swiss prot sort of subset of proteins from, I think, UniRef. Yeah, I think that's the sort of larger set. And yeah, we had to do some substantial filtering on this synthetic data, so I will describe that now. So basically, we started with over 500,000 AlphaFold-2 predictions of sequences from this Swiss prot collection. And then the first thing we do is we filter on essentially global confidence. And yeah, so we want only high confidence predictions by the model. That's this PLDDT metric. We also want to mask the loss for residues with a low confidence, because it's possible that globally the structure might have high confidence, but there could be sort of disordered regions of low confidence, and we don't want to backprop through the loss from those residues. And so we mask things that have confidence that's low. And then the last thing we do is we found that even though we sort of filter on global confidence and we do this residue masking, there were still high confidence, but sort of not good examples in our training data that introduce noise. And so, yeah, these were needed to be filtered. So we learned a model to do sort of a prediction of good, bad, and then we're able to filter it out. So at the end of this filtering, we're left with 156,000 synthetic structures, and then combine that with the ones roughly 30K from PDB that we used, and that's this larger data set. So as we'll see in a little bit, our absolute best results were still with PDB only, but the difference was not huge, and we're still investigating why that is and how we can improve performance using this synthetic data as well.
For fine-tuning, so we did something similar as done previously in FrameFlow. Essentially, we augment the data. We take the one from PDB or synthetic, so in practice PDB, and we augment it with a random motif mask. So we say, okay, part of this chain, I'm gonna fix it, and the rest I'll try to design it to mimic this sort of motif scaffolding task. And so we start with our protein. We sample a motif and a scaffold, mask the input. So now I want to fix my motif and flow my scaffold, or design the scaffold. Give this into our model, and keep this in mind. In this case, we always have also the sequence of the motif. And then we have a predicted vector field that we only applied to the scaffold. And so and then the loss, of course. But so on the right, we see like this design, so where the motif is fixed and the scaffold flow along the path. So we fine-tune for about 300K steps, while other methods usually train entirely with this kind of augmented data. In RF diffusion, they did this 80% of the time. The main challenge there was really the centering, the discrepancy sometimes between training and inference. So we had to iterate a few times on this to get it right. Okay, so a quick recap. So now, yeah, at the beginning, we talked about like these ML landscape in protein design that were really like specific to some modalities. And now what we see more and more is combining these modality for folding, for generation, and inverse folding. So for example, AlphaFold-3, Rosetta Fold, or Atom, they use sequence, molecule, DNA information, and they fold it into structure. And generation, now more and more people use sequence and structure to generate structure. So our model, we also see Genie2 does something similar. And other models just use sequence and structure, and other modalities like template, and design the sequence and structure as well. So ESM3 does this, Multiflow, another model that does something similar. So now we'll follow with the experiments. Yeah. So before really presenting the results on unconditional generation, we'll just quickly review how do we evaluate the generated protein. So a first criterion is the designability of the generated protein. We start by generating 50 protein of length like 100 to 300. We then inverse fold this protein. So we get the backbone, we apply protein MPNN 8 times to get 8 sequences per structure. We then refold these sequences with ESM fold and compute the RMSD between the generated structure and the refolded structure. The idea is that if the two agrees, then it's designable. And the threshold is 2.0 Armstrong. So we don't only care about designability, but we want to make sure we design proteins that are more diverse and novel as well. So for diversity, we compute the TM score, so a template modeling score between our generated set and our reference set in the PDB dataset. We also compute the number of clusters proportional to the number of generation. So like how many clusters did we generate actually? And so we want to make sure that our generated set is farther away than a reference set in the PDB dataset. For novelty, we use the average max TM score between the generated and reference set for the protein that are designable and novel.
Okay, so given those 3 criteria for unconditional generation, basically the high level conclusion is that, you know, if you do all of these things, you train this architecture with this data, it's pretty good. Yeah, so as you can see, it's the chart sort of shows that we're roughly, I mean, we're basically us, RF diffusion, Genie as well, we're more or less maxing out the designability metric, which is a substantial improvement from FoldFlow as well. So that's sort of puts us on par with kind of these very designable models. Where we see a pretty significant improvement over previous models is in the novelty and diversity categories. And there we see that, yeah, we're significantly better than FoldFlow as well as RF diffusion in novelty and diversity, and you know, even slightly better than Genie 2, which was a concurrent work. So, you know, to be honest, we don't completely understand why where this improvement in novelty and diversity comes from. We have some hypotheses. One of them is the inclusion of sequence data. That seems pretty, so this is a trend that I believe the multi-flow paper also observed, where adding sequence data improved novelty and diversity. And another intuitive way to think about it is that to the extent that the information in ESM2 is distilled into the weights of FoldFlow-2, because remember, we don't have the ESM2 representations at unconditional generation because we completely mask the sequence. But during training, you know, 50% of the time, those representations are seen. And so it's possible or intuitive maybe that some of that information is being distilled into the weights of the FoldFlow-2 network. And ESM2 has seen many orders of magnitude more proteins than we have structures for. The other thing that could be driving it, it's not exclusive with the previous one, is the inclusion of the evoformer. Yeah, basically, I think Genie 2 also uses an evoformer and shows a similar improvement in performance. And it could just be that this architecture, which wasn't in FoldFlow 1, you know, is able to more efficiently process the protein information and basically just learn a better fit to the data. So these are some ongoing hypotheses for why, but we are quite pleased with the performance that we get. There's another way to measure the performance, which sort of looks at the relationship between novelty and designability. Essentially, at one extreme, it's possible to generate very novel proteins, but that are not designable at all because they're just sort of so crazy that they could never exist in nature. And then at the other extreme, you could imagine a model that regurgitates its training data, which of course is designable because they're naturally observed structures. But that's also not very useful because you can't get any new generations out of that model. And so between these two extremes, we basically plot sort of the various levels of designability given a level of novelty, and sort of up and to the left is better. And we see that FoldFlow-2 substantially outperforms the previous sort of best, which would have been RF diffusion, and even Genie 2, which is again a concurrent work, we're slightly better as well on this metric. So yeah, I think it's a pretty powerful setup. We did some investigation into some of the hyperparameters that were found to be important in FoldFlow-1. So one of them is the number of integration steps that you take during inference. And so there's a sort of inverse relationship between the designability and novelty as a function of the number of Euler steps. And so fewer steps leads to more designable structures, but also less novel ones. And so I think this was observed in FoldFlow-1, and it's good that we're reproducing it. The other thing that we observed is the importance of this inference annealing trick, which was pretty important to the FoldFlow-1 success. And that seems to remain true for FoldFlow-2. And so it's likely that this general property of sort of protein flow models. Yeah, so those are some results from the unconditional generation. Another way you can characterize the diversity is by looking at the proportion of secondary structures in a given generation. So the three types of secondary structures are in blue, green, and red in the top right. And you can plot those on this ternary plot. As you see, this is the sort of training distribution. So a good model would fit this distribution accurately. And so what you see in FoldFlow-1, it suffered pretty significantly from mode collapse, where almost all of the generations were helices. For RF diffusion, it is substantially better at capturing the modes, but doesn't really capture the spread of the distribution as well. For FoldFlow-2, we see a significantly, or at least substantially better improvement in this sort of spread capture of the distribution relative to the training data set. And then we also experimented with a setup where you can further fine-tune using this reinforced fine-tuning objective to specifically encourage the model to produce diverse secondary structures. And so if you use this fine-tuning objective, you can really steer the model to sort of respect this the scalar reward that depends on the diversity of secondary structures. So that's a pretty interesting result as well. And then, yeah, we also, as I said, experimented with synthetic data. And like I said, it's good, but not quite as good as PDB. So most of our sort of main results in the paper use the PDB data set, but you know, it's the differences are pretty marginal, and we're still working to improve that recipe.
So I'll quickly go over this in the interest of time, but here we just wanted to sanity check that we indeed like take the sequence representation into our account. And so we just evaluate on the folding test since we mask it 50% of the time. So we start from a test set of like a bit less than 300 sequences, fold them and compute the RMSD between the folded and ground truth one. And so we see like it's not as good as ESM fold, probably AlphaFold and other like folding model, but we do see a reasonable prediction, and it does better than other multimodal or other models that can perform multitask. And we also experiment with scaling, so increasing the number of parameters, and we do see every time an improvement by increasing the number. And just as a reminder, like ESM fold, I think they use something like 40 blocks of evoformer, while in this one we used 4. So there's definitely like a way to improve it, although it's not the main focus in this paper.
Yeah, okay, so I'll quickly explain how we evaluate motif scaffolding because it's a little complicated. Basically in our paper, we have this table with sort of 3 columns for criteria, as well as an overall designable criterion. I'm going to explain how those are derived individually. So basically for motif scaffolding, right, you start with a motif which has a structure and a sequence associated to it, and then you sample the scaffold sort of noise for that, and then pass all of that into FoldFlow-2. FoldFlow-2 then generates essentially a scaffold structure, and the motif is kept fixed, and so you have this full generated backbone that has a motif region and a scaffold region. And you can take all of that information as well as the sequence, and you pass it into Protein MPNN, and you ask Protein MPNN to design a sequence for the scaffold while keeping the motif sequence fixed. And then you take that predicted sequence, you pass it to ESMFold. This is actually a notable difference. We used ESMFold instead of AlphaFold-2 in our eval pipeline. So it's not directly comparable with sort of the original RF diffusion numbers, but we recomputed them. And then, yeah, you have this refolded backbone, which has a sort of motif region and a scaffold region. And then the first thing you want to know is, so this first column, is the overall generated backbone sort of valid or designable? And then if that's true, you also want to know, does the motif sort of, is that respected in the generation? And so that has a stricter criterion for designability. And then lastly, you want to know, is the scaffold itself designable as well? And so, if you have all of these criteria satisfied, then you say that the generation or the motif scaffold is designable. And so given that evaluation procedure, with the generic pseudo-label fine-tuning that Guillaume described earlier, we see the results are basically good, but not great, in the sense that both FoldFlow-2 and RF diffusion are able to solve sort of all 24 examples, basically solve meaning if any of the 100 generations are valid. This is the original criterion used from RF diffusion. However, if you dig into it as we did, it's clear that RF diffusion has a significantly higher success rate than FoldFlow-2, and so clearly there's still something going on there. One of the things, as we mentioned earlier, is that RF diffusion, as well as Genie-2, pre-train with some percentage of motif scaffolding as a task. RF diffusion, it was 80%. Genie-2 is actually 100%, and they were still able to do unconditional generation extremely well. So clearly, this is something that we'd want to look more into in the future. We also looked into motif scaffolding in the situation where you have sort of in-domain knowledge or in-domain data to fine-tune on. And so the domain is the VHH nanobody, which is a family of proteins. And they have this known region called the CDR, or complementary determining region, that we wanted to try to see if we could generate scaffolding for. The CDR this problem is hard because the CDR regions are kind of loopy and flexible, and CDRs are known to be important for biological function. So it's a reasonable thing to investigate. Yeah, basically we constructed this data set using there's a train set and a test set. And yeah, basically we fine-tuned on the train set and then evaluated both RF diffusion and FoldFlow-2 on the test set, and found that when you can fine-tune with in-domain knowledge, we're able to outperform RF diffusion, which is a very powerful sort of baseline model. So this is a pretty interesting result around how can we steer the model to do useful things.
Yeah, so we had this last experiment, very preliminary results where we wanted to look at zero-shot equilibrium conformation. So in this case, we want to look at the stable distribution of a protein, so of the possible conformations. Usually this is predicted with like molecular dynamics simulations, and we look at all the possible folds of that protein. So here for one sequence, let's say we generate 100 structure and we look at the variation at the atoms. And we see that, so on the left, we have the ground truth. This is like the distributions of conformation. In the middle, we have ESM flow trained specifically with molecular dynamic and FoldFlow-2. So we see we're able to picture a broad range of diversity of conformation. And this task was motivated for us in order to design potential binders to flexible targets. And so in this case, we would also fold the target and be able to design binder for diverse conformations. This is part of our next steps. So in conclusion, the next step in takeaways, we introduced a new sequence condition model with improved designability and diversity. We solved all the RF diffusion scaffold benchmark, and we introduced a new scaffolding data set. We compare also with RF diffusion on this one. And next, one of the things that interests us is the designing of binder with fixing or folding the target. So on the right, we have a GIF of like these sort of trajectories, what you would fold and design at the same time. And we want to validate FoldFlow-2 in wet lab experiments. Next. So to conclude, just want to recall that this was a very big team effort at Dreamfold. Everyone was really involved in experiments, design the model, a lot of components. And you can find more experiments in the paper, more results, and on a blog post as well that we've written. Thank you.
Thank you very much, Guillaume and James. So we have time for some questions, and I have a few already in the chat, and you're more than welcome to add some more. So maybe first question, can you explain what happens after the geometric decoder? I don't know which slide it refers to.
Maybe I'll go back quickly. So the geometric decoder. So maybe I can take it, but so the output of these IPA transformer is always an updated rigid representation of like translation and rotation. You also have an edge and pair representation, but for the decoder, we only use the updated rigid representation. At inference, so we use directly the rigid, or at training, we compute the vector field given this rigid that we use then to update our gradient from the loss. Does this answer the questions?
Well, looks like, I don't know, I'm biased because I know.
Okay.
Then another question about the architecture. When encoding an IPA, do you represent proteins by C alpha positions only, or do you use frames?
So I don't know.
Yeah, go for it.
Okay, so we use both essentially. So this comes from the representation that we designed in the beginning and that other people use as well. So we split into a translation and a rotation part. And the rotation is the C alpha, and the rotation, the translation is the C alpha, and the rotation gives you the position of the N and C. And so yeah, it's really the frame that we use as input in the structure encoder.
Thank you. And then, well, from the same participant, instead of skip connections, did you try just passing the time embedding directly to the decoder?
That's a good suggestion, yeah. I don't recall if we did try this, but yeah, this is definitely something we could experiment and test.
So yeah, the time and position embeddings are sort of all combined together at the start in the encoder. And we do want the position embeddings to be available to the Evo former and to the sort of the fusion trunk. So yeah, I think we could definitely try the time embedding, but I think having it sort of present at the inputs as well as sort of in the outputs is quite important. So yeah, I think that's a good idea though.
Thank you. Another question again about the model. When training the model that predicts whether a synthetic AlphaFold-2 structure is good or bad, even for high global confidence predictions, what's the ground truth label here? Some reference-free metrics, maybe.
Yeah, that was, I think, sort of we annotated a set of proteins that I think we annotated with designability itself as the sort of criterion, and then we're able to learn a model that, using some fairly simple structural features, was able to predict if the synthetic structures would be designable pretty reliably.
Thanks. What kind of novel proteins can you design with FoldFlow-2 based on the results?
So what do you
Yes.
novel, what kind, what is that?
Question is probably a lot talks about families of proteins, so I don't know what it's antibodies. Well, I'm interpreting it myself, but I don't
see.
know.
Guillaume, do you want to talk about?
Yeah,
I mean,
it's a good question.
all kinds.
We don't have a direct answer. Yeah, exactly. I don't think we have a direct answer to this. We looked at the number of clusters that are different, whether it's different to a reference set, but not yet so much as like what kind of protein they are. This is something we investigate.
Yeah, I think like it seems so things like antibodies are likely going to be hard for these like models that emphasize structure so much, just because the structure of an antibody, which has lots of flexible regions, is not sort of uniquely determined, and so that makes it probably a lot more difficult to train and learn to generate good ones. The conformation prediction stuff that we were looking at, as well as the ability to sort of partially fold binders or like partially fold these binder target pairs, is maybe would make those challenging targets slightly more approachable for our models. But the things that are promising are sort of short and relatively structurally stable proteins like mini binders or things like that.
So thank you. Another question. I know that we're almost at time, so probably will take just a few. The model uses a modest amount of compute, 2 GPUs for 4 days. Do you think the results would benefit from scaling, or are you not seeing gains there?
The results do benefit from scaling. Yes, this has been, so, why would we grab, a bunch of structures from synthetic data if we're not planning to scale the model? Well, we're working on scaling actively. Yeah, so you do see improvements with scale. I mean, we kind of even have a teaser of that in the folding section, right? Where it's relatively easy to evaluate sort of good folds from bad folds, and you see that as the model increases in size, the folding capabilities increase substantially.
Thank you. And can you say more about data splitting for the conditional tasks?
So the folding task, I assume?
Yeah, I think so.
Yeah, so we took the test set from Multiflow that we then had to take a subset, because part of our training set was in their test set. So then it's around 300 protein. And so it's a test set for Multiflow, ESM, Fold, and ours also. And that was the split essentially. I think in the original paper, Multiflow, they have a bit more, but we had to remove a few for us because we trained on.
Thank you. And maybe last one, did you try a stochastic integration scheme, or is everything shown here done with the ODE Euler integration?
So everything is, oh yeah, please.
Well, no, go ahead.
I was about to say, yeah, here everything is Euler, but we do have an option like in FoldFlow-1, we experiment with also a stochastic path. I think maybe in the appendix, or remember somewhere we try it a bit, but it wasn't it's something we need to continue exploring, I suppose.
Yeah, basically, yes, we did. The results are in the paper. The best results were with the optimal transport variety of flow matching. And also I think in the FoldFlow-1 paper, you saw that the stochastic flow matching was really able to help on, I think, novelty and diversity. But for us, we were able to achieve even better results without using the stochastic flow matching. And so we don't really have to give up anything in terms of designability, but we get strong novelty and diversity. So that's why the main results don't use it.
I see. Well, thank you very much. I think that's all. It was great having you here. And as usual, the lecture will be recorded. It was recorded and it will be published on YouTube after some editing and approval from your side. So again, thanks everyone for joining. It was a pleasure as usual.
Great. Thank you very much.
Thanks a lot. Yep.
Thanks.